I'm James McCarroll. I'm the current dean of the Skaggs School of Pharmacy and Pharmaceutical Sciences, but we're here to honor the founding dean of this school, Palmer Taylor. So, sure, you can applaud. So let me briefly tell you about Palmer. Actually, the story begins in 1896 in Wisconsin. Now, if you're in Wisconsin today, you'd probably be cross-country skiing, but you're not. You're here. But in 1896, there was a drugstore in Wisconsin, a drugstore that was actually founded by Palmer's family. It was Taylor Drugs. So this whole unit, this whole history of pharmaceutical sciences and pharmacy actually goes all the way back to the 19th century. And I think all of us will learn today about the contributions that Palmer has made to science, which are enormous. So many really seminal publications in areas that include the acetylcholine receptor, acetylcholine esterase. So when you think about these acetylcholine esterase, for example, the target of toxins, so research on toxins as well as research on signaling within the central nervous system. So a phenomenal diversity of scientific contributions that Palmer has made. And today and tomorrow we'll hear from many renowned scientists from around the world who in some cases are even former students and postdoctoral fellows of Palmer or have been recruited here to continue his legacy. But I'd say that in terms of his legacy and collaboration, perhaps the most important collaboration that started was in 1963 when he married Susan Taylor.
65.
65. Okay. So 63 would be before the Beatles. 65 would be after Hard Day's Night, which came out in 1964, correct? Right. And somewhere up here will flash a picture of Palmer and Susan in Palmer's cherry red 1965 Volkswagen Beetle, which of course has an air-cooled engine that I'd love to work on, but I can't. We're going to follow Palmer's contributions to science by hearing from a number of people, but I think also you should be aware that you are sitting in an auditorium in a building that would have not existed had it not be for Palmer. And I must say that for myself, I have the easiest job in the world. Coming here as dean was simple because Palmer had set the stage. He had made the foundation. He had recruited a top-flight faculty to this school. He had already graduated about 290 students from this school with pharmacy doctorates. So the people that he brought here are people that help me every day. And it's amazing because I continually think, wow, I really don't have to do very much because it's all been done by him. So I think he deserves immense credit for having the vision that started this school. And this school is very special if you don't know about it. This school is special because the graduates of this school are not just graduates with a Doctor of Pharmacy degree. They understand the science behind the way drugs work. So it's a combination of research and pharmaceutical sciences as well as drug formulation and delivery. So I'm looking forward to hearing from everyone today, and I'd now like to turn the podium over to our Chancellor, Pradeep Khosla, who is the 8th Chancellor of our university. And some of you may not realize this, but he's also a renowned electrical and computer engineer. And I mention this because if you have any problems in your house, if your garage door won't open, toaster won't work, you know who to talk to, Pradeep.
Thank you, Jim, and good morning and welcome everybody. Welcome to sunny San Diego. Well, if you've not seen the sun yet, you will. Trust me, it shows up. It's only a matter of time. I see, as I was coming here and thinking about what is it that I will say, I was reading the title of the symposium, and I realized that there are a couple of words missing. So the symposium says something like, in honor of Palmer Taylor, scholar, scientist, mentor, dean. And I said, I know a little bit about the man, but I know nothing about his science. I don't understand his science. And I said, maybe I should read the titles. And then I thought maybe the symposium was in Latin. But no, it was Latin to me. I realize the symposium is in English. The titles are in English. But Palmer is an amazing person. I think the words that I would describe him with as the chancellor that were not there were visionary and leader. In academia, we have a lot of great scientists. That's what makes great institutions. But great institutions also have a few great leaders. And I think Palmer is in that category. Palmer's vision started long before the School of Pharmacy was created. His vision in his own science was truly world-changing. And I'm looking at his resume, and I said, you know what, I'm sure he's won all the awards, but if there's an award he's not won, it's not because he doesn't deserve it. It's because he's such a giant in his field, people just assume he has won everything, so nobody nominates him for anything else now, because he's a very big giant in his field. So anyway, Palmer, so thank you for being at UC San Diego. Thank you for all the commitment that you've made to UC San Diego. And I can tell you, so when I came here and I'm looking at what a great institution this is, and how are we going to make this institution even greater and position it in a sustainable manner for sustainable greatness, I realized that the School of Pharmacy and Pharmaceutical Sciences was going to be at the center of just about everything we do. And in our strategic plan, one of the strategies we identified was vertically integrating basic discoveries up to therapies. And you can imagine that in the School of Pharmacy and Pharmaceutical Sciences, there are not just basic discoveries in chemistry, but they're also building the drugs and the therapies. In our School of Biological Sciences, Division of Biological Sciences, the same thing is happening. And Scripps Institution of Oceanography. So if I look at UC San Diego overall, this is the school that connects all of our components together, and this is the school that propels us into the outer stratosphere that we actually believe we deserve to be in. This is also the school that in less than 10 years was in top 10 in the country. And that's an amazing feat. I don't know of any other institution where you can create a department or a school and catapult it to top 10 when there are 131 other schools in the country. So today there are 132 schools in the country. And again, yet one more time, this is a school, thanks again to the founding faculty, the quality of faculty that Palmer hired, his vision, has a 100% success rate in its graduates when they pass their exams. So I could not say enough good things about this school. I could not tell you how important this is, whether in integrating our Scripps Oceanography with drug discovery, integrating pharmacology with drug discovery, biology with drug discovery or building new drugs. But this is the school that's going to change the world. And I know that each one of you who's traveled from all over the world, who are also leaders and leading scientists in your fields, believe that yours is a school that's going to change the world. And I guarantee you that is true too. But I'm hoping that this school would be in the same company as your school, and together we'll change the world, because the world needs everything that you all do. So Palmer, I know this is to honor you, but let me tell you from my perspective, you honor us by being here. So thank you very much for your leadership, and welcome everybody.
So I'm Andy McCam, and I'm very honored and pleased to be part of this program as chair for part of the first session. I'm one of a number of faculty members that was recruited here by Palmer and Susan. Palmer is very cagey in recruiting new faculty members. He knows how to pluck all the right strings. I remember he sent me in an early packet the curriculum vitae for one Roger Chen. Said, he might like to be a colleague of this fellow. And I looked over that CV. I said, yeah, he's a pretty good guy, you know. It's a good colleague to have. And came out here. I had worked here as a high school student in 1964 at the Scripps Institution of Oceanography. So Palmer took me up on a bluff to overlook the SIO and summoned all the reminiscences of fond times. And lo and behold, here I am. And I think a number of us can tell similar stories. My job here is just to basically keep the first three speakers on time. I'm sharing the chairing duties with Bill Catterall, who's our first speaker. Bill's from the University of Washington. Washington and UCSD have a lot of common commerce in pharmacology. There's a lot of work that's in the kinase field that has roots there, a lot of work in ion channels and other receptors. And so it's a very great pleasure to introduce Bill Catterall as our first speaker.
So thank you very much, Andy. It's a special pleasure for me to give the first talk in this really outstanding symposium in honor of Palmer Taylor. So I'd like to tell you about our recent work on structure and function of voltage-gated sodium and calcium channels at the atomic level. But before I do that, I'd like to say a few words about Palmer. Who's really a world leader in both research and in academic administration in pharmacology. In fact, I'd say he has really an unparalleled series of accomplishments combined in those two areas. So in research, as you've heard, he's been a world leader in acetylcholinesterase, a recent crystal structure shown in the inset here. Looking back, his cloning and primary structure of acetylcholinesterase and determining the molecular basis for the different isoforms and how they are distributed in tissues is really pathfinding at the time in the 80s, and much more work on the structural basis for catalysis, pharmacology, and more for the acetylcholinesterase family of enzymes. He's also worked on acetylcholine receptors and done seminal work in that area, including work on the mechanism of activation and desensitization that Steve Sine will enlarge on in his talk later, and much work on the structural basis for the pharmacology of nicotinic acetylcholine receptors as well. And as you've heard eloquently from our two previous introductory speakers, Palmer has had an amazing impact in academic administration as founding chair of the Department of Pharmacology here now, fourth ranked worldwide by U.S. News and World Report and under the leadership of Joan Hiller Brown, and founding dean of the School of Pharmacy and this magnificent facility. So there's much to celebrate. Congratulations, Palmer. Let me turn now to my main topic, which is sodium and calcium channels. We've known for a long time the physiological role of these two key electrical signaling proteins. Hodgkin and Huxley showed in their pioneering work in 1952 that sodium channels are responsible for the arising phase of the action potential initiated at the nerve cell body. They activate, and then within a millisecond or so, they inactivate through a fast inactivation process. The action potential proceeds down the axon, reaches the presynaptic nerve terminal, and activates a calcium current. And voltage-gated calcium channels initiate excitation secretion coupling and exocytosis of neurotransmitter at the nerve terminal. Neurotransmitters move across and bind to neurotransmitter receptors, and those will be the topics of the following talks in our symposium this morning. So when I began my lab in Seattle in 1977, sort of the same era that Palmer was building the division of pharmacology here, a lot was known about how sodium and calcium channels work in a physiological setting and about their electrophysiological properties. But really there wasn't anything known about the molecules themselves. So that became our first main focus of interest. We took advantage of neurotoxins that are active at sodium and calcium channels, block their function in one way or another, and in that way immobilize prey. You'll hear a lot about neurotoxins from other people, including Toto Oliveira tonight, and I've just shown you here a few of the handsome creatures that make these nasty toxins. So frogs that make batrachotoxins, scorpions that make peptides, scorpion toxins, cone snails that Toto will tell us about, and a tarantula. So we used particularly scorpion toxins and saxitoxin, but also many of these others, to identify the sodium channel protein in brain, purify it, and characterize its molecular composition. As you can see, it is a complex of a large alpha subunit of about 260,000 Daltons in association with smaller beta subunits. We found from biochemical experiments that the alpha subunit is a transmembrane protein, contains the pore and the toxin binding sites. I'm going to... All that scraping is my badge. Scraping on the microphone. Hopefully that will quiet that down. Purified sodium channels we found could be reconstituted into planar bilayers, and we could record the activity of single purified voltage-gated sodium channels and convince ourselves we'd really purified the right thing. Some years later, we used dihydropyridine calcium channel blockers to isolate the calcium channel. It has a alpha-1 subunit that's a cousin of the sodium channel, as you'll see, and it's associated with a different set of auxiliary subunits. Sodium and calcium channels are the founding members of a large superfamily of membrane ion channels. And the voltage-gated ion channel superfamily includes these two proteins, 10 genes for each, potassium channels, trip channels, and in total 143 different voltage-gated ion channels and relatives. I want to call your attention to the fact that sodium and calcium channels, according to this dendrogram, are predicted to have a common ancestor from which they both evolved, and we believe that ancestor is a bacterial sodium channel that I'll introduce to you in the main part of my talk. So, sodium channels were first cloned in the laboratory of Shusaku Numa in Kyoto, and the left part of this slide shows a folding diagram of a mammalian voltage-gated sodium channel. They're proteins of about 2,000 amino acids in length, the alpha subunit, organized in 4 repeated domains. Each domain has 6 transmembrane segments, and those 6 transmembrane segments divide into 2 functional modules: a voltage sensing module, S1 through S4, and a pore forming module, S5 and S6, and the P-loop in between. In addition, and very important for sodium channel function in mammalian tissues, sodium channels have a fast inactivation gate that's formed by this intracellular loop between domains 3 and 4. Now, if you were interested in the structural biology of sodium channels, and you looked at this structure, you'd say, wow, that's not a very good thing to work on. It's about the worst thing you can imagine for X-ray crystallography. 24 transmembrane segments, large and unstructured intracellular loops, and so on. So the discovery by David Clapham and his colleagues of the bacterial family of sodium channels, first one called Natchback, was really important. You can see that this nice little molecule is analogous in structure and, as it turns out, in function to one of the domains of a mammalian sodium or calcium channel, and it functions as a homotetramer. So this has turned out to be an ideal substrate for structural biology and detailed structure-function analysis, and that's kept us busy for the last several years. Still getting noise from the microphone. I'm not exactly sure where it's coming from. So we cloned several of these bacterial sodium channels and studied them biochemically to find one that was ideal for crystallography, and we were able to determine the structure of the voltage-gated sodium channel from Arcobacter butzleri, or NAVAB, at 2.7 angstroms resolution. Our best structures are of a single residue mutation, I217C, and they extend to 2.7, or in favorable cases, 2.5 angstroms resolution. So you can see an early structure here looking down on the channel as sodium ion would. You can see the pore-forming modules in blue, the central pore here, and the 4 voltage sensing modules in warmer colors displayed around the periphery in an almost symmetric array. If we look at the structure from the side, we again see the central pore-forming region in blue, the 4 voltage sensors that are arrayed around the periphery, the key S4 S5 linker in red, the covalent connection between the voltage sensor and the pore. Looking a little more closely at the architecture of the pore, on the left side, you see 2 subunits, 2 pore-forming modules on the side of the central pore. The S5, S6 segments and the P-loop are illustrated. And you can see a sodium ion first enters an extracellular funnel, passes through a narrow ion selectivity filter, moves into a larger water-filled central cavity, and if the pore were open, it would exit through the activation gate. But the activation gate is tightly closed in this structure, and so this is a closed pore conformation. You can see how tightly closed here the side chains of methionine 221 completely occlude the opening of the pore, so no sodium ions can move. So in the next 20 minutes or so, I'd like to take you through each step in the functional cycle of sodium channels and give you a little insight as to how that we think that works at the structural basis. And so the first is voltage sensing. So perhaps the most exceptional property of the voltage-gated ion channels is their voltage sensitivity. Many plasma membrane proteins feel or exist in the same membrane potential as a sodium or calcium channel, but aren't affected by it at all. Whereas sodium, calcium, and potassium channels activate and inactivate with exquisite sensitivity to voltage. So how does voltage sensing work? This shows the crystal structure of the NAVAB voltage sensor. It has 4 transmembrane segments, same 4 transmembrane segments as I showed you before. So starting from here, the S1, S2, S3, and S4 transmembrane segments form a helical bundle with an aqueous cleft between these 2 transmembrane helical loops. The key functional elements in the voltage sensor are the gating charges. 4 arginine gating charges called R1 through R4 in this slide. And they're arrayed across the membrane, 1, 2, 3, 4. You can see their side chains along here. These are the charges that sense the membrane field and changes in the membrane field. In this structure, 3 of these charges are outside of a key point, the hydrophobic constriction site here, and 1 of them is inside. The hydrophobic constriction site is kind of like a rubber seal in the voltage sensor. It keeps water and ions from moving from inside to outside or vice versa. On the outside of the hydrophobic constriction site is the extracellular negative cluster, 2 negatively charged side chains of glutamate or aspartate residues which interact in the structure with the gating charges. On the inside of the hydrophobic constriction site is the intracellular negative cluster, INC, and its negative side chains interact with the R4 gating charge on the inside of the hydrophobic constriction site. So this is an activated conformation of the voltage sensor because 3 of the gating charges are outside the hydrophobic constriction site. So our crystal structure has captured NAVAB in a pre-open state with activated voltage sensors, but pore not yet open. In normal physiology, this state would be short-lived and the pore would snap open within a millisecond. But we think we've trapped the channel in this state by the isoleucine 217C mutation that I mentioned previously. Now, we think voltage sensors function by moving, pardon me, lost my button here, moving these gating charges back and forth through the hydrophobic constriction site in response to the electric field, changes in the electric field, and in that way initiate a conformational change that opens the pore. And this is actually a long-standing idea. In fact, when the first sequence of a voltage sensor was published in the 1980s, I put forward a model called the sliding helix model in kind of cartoon form. And Palmer thought it was a cool idea. He actually put it on the cover of this wonderful textbook from about 1990. And this is the cartoon form of the sliding helix model, which we have now made into a structural form in more modern style. But I really appreciated Palmer's support of our model at that time. It's been very controversial, Palmer, but you were on the right side early. So here's our current model, which is structure-based. The key thing is to know what the structure of the resting state is. We have the structure of the activated state, as I showed you, and we have that for potassium channels from Rod McKinnon's work as well. So with David Baker, a leader in molecular modeling, we created a model of the resting state of the voltage sensor, which is shown here. And you can tell it's a resting state because of these 4 gating charges. They're now little blue balls here. 3 of them are on the inside of the hydrophobic constriction site, which is just here, the gray residues. Here's the extracellular negative cluster and the intracellular negative cluster. This is a little movie, and when I run it, we'll go from the resting to the activated state of the voltage sensor. As 4 gating charges will move out, the other transmembrane segments will make accommodating rotational moves. They pull on the S4, S5 linker, and they pull open the activation gate, which is right here in the structure. So I'll run it and then come back to it one more time. So like that. It didn't run. Hello. There we go. Thank goodness it ran. So you see, S4 segments move out. Conformational change takes place in the voltage sensor, pulls here, and opens there. So one more time. Now, we've done a lot of experiments to confirm this model, and people working in potassium channels have also contributed greatly in this area. I don't really have time to go through that with you this morning, so let me just tell you the type of experiment that we found most convincing. So, among its other gifts to science, NAVAB is cysteine-free. So we place cysteine residues in position of one of the gating charges, and in the position of either the extracellular negative cluster or the intracellular negative cluster. And we followed state-dependent disulfide bond formation as in either the resting or the activated states. And for example, if we put a cysteine here and a cysteine here, it does not form a disulfide bond in the resting state. But as soon as we activate the channel, they come close together right there, and they form a disulfide bond in less than a millisecond. So this kind of information from our lab and much work on potassium channels led to a consensus article in 2012 from many of the leaders in this field. The emerging consensus is that this type of model, initiated as a sliding helix model long ago, does indeed give a good view of voltage sensor function. And so I'll just run it one more time. Oops, sorry, come back. I'll just run it one more time. And I want to give credit to my graduate student, Paul DeCon, who really did fantastic work on disulfide locking in the papers that are listed there. So the second most important thing the sodium channel does after activating in response to voltage is conducting sodium rapidly and selectively. So how does that work? So you've already seen the pore, and I called your attention to the ion selectivity filter, the narrow spot that sodium ions have to go through. So here is the outer edge of the ion selectivity filter as a sodium ion would see it coming down into the pore. It's formed by the side chains of 4 glutamate residues, E177 for those who can remember sequence numbers. And this is a very high concentration of negative charge. We call it the high field strength site. This site seems to be essential for selectivity for sodium. If we look at the selectivity filter from the side, there are actually 3 interaction sites for sodium. The E177 high field strength site here. In the center of the selectivity filter, central site, formed by the backbone carbonyls of leucine 176. And an inner site formed by the backbone carbonyls of threonine 175. And after sodium passes through the selectivity filter, it moves into the large cavity that is water-filled and then on into the cytoplasm. Importantly, the selectivity filter is quite wide, as the slide says, 4.6 angstroms. It's sized to accommodate a hydrated sodium ion. It's far bigger than a bare sodium ion, which is only 2 angstroms in diameter. So we believe, on this basis, and also on the basis of observing water in the right positions in the selectivity filter, that sodium is conducted as a hydrated cation with both planar and axial waters of hydration as it moves through the pore. On the right side, we've shown these liganding groups in red, so the high field strength, central and inner sites, and then this is a little animation that will give you a sense of the three-dimensional view. As sodium comes through, it interacts first here, and then here, and then here, and on in, and it stays hydrated through that whole process. This selectivity process is quite different from potassium channels, previously described in beautiful work by Rod McKinnon and his colleagues. In potassium channels, the selectivity filter is much narrower, as illustrated in blue here, than the sodium channel selectivity filter, which is in yellow. Indeed, the backbone will sit inside the sodium channel selectivity filter. And that's because a potassium ion is completely dehydrated during conductance. It binds to 4 sites, 1, 2, 3, 4, as it moves into the cell. All of these are formed by backbone carbonyls. There are no negatively charged side chains involved in potassium conductance. So the sodium channel selectivity process is chemically distinct in 2 important ways. Interactions with charges are necessary to partially dehydrate and select the sodium ion. And the sodium ion is conducted in a hydrated form with its inner shell of waters largely or completely intact. As I mentioned on the first slide with the Hodgkin-Huxley citation on it, sodium channels activate and then within a millisecond they inactivate. And this fast inactivation process is crucial for sodium channel function in nerve and muscle, where repeated action potentials require inactivation, repolarization, and then the next action potential to be fired. So how does inactivation work? Well, for fast inactivation, we have to jump back in evolution or forward in evolution to the mammalian sodium channel, because as I told you, the fast inactivation gate is here in this intracellular linker between domains 3 and 4, and the bacterial sodium channels have no such structure, right? Because they just are a tetramer of 4 separate subunits. So in the vertebrate channel, this inactivation gate folds into the pore during the inactivation process, binds there through interaction with a trio of hydrophobic residues, isoleucine, phenylalanine, methionine. We know the structure of that little piece of the mammalian channel from NMR analysis with Rachel Clevitt at the University of Washington. And as shown here, there's a rigid alpha helix preceded by 2 loops on which the key isoleucine, phenylalanine, methionine, and a neighboring threonine are arrayed. And we think this is just plunges in and blocks and binds and blocks the intracellular mouth of the pore during activation. During fast inactivation. So here we have another movie. It's a kind of a combination of science and art in this movie. The science is that this is the same core structure that I showed you previously, different colors, of the bacterial sodium channel, 1 voltage sensing domain, a complete pore module, some sodium ions outside. So it will run just like before. The art is that we have, shamelessly I have to say, taken the mammalian inactivation gate and pasted it onto the bacterial channel to give you a sense of how we think this works. It's actually great in medical school lectures too. So here we go. And you can see as before, come back. You can see as before the voltage gating charges move out, voltage sensor pulls, pore opens, sodium comes in, inactivation gate closes. So every time your nerve generates an action potential or your heart beats, your sodium channels are doing that. You can think about that. Now, sodium channels have a second form of inactivation, slow inactivation. Slow inactivation was discovered in the squid giant axon, actually, not by Hodgkin and Huxley. And it takes place in vertebrate sodium channels on the 100 millisecond time scale. It's important in mammalian nerve and muscle to protect against too many action potentials being fired, so it is engaged in long trains of action potentials. So the bacterial channel NAVAB has a similar slow inactivation process. It's faster slow inactivation than in mammals, if I can say that. So you see it here. During a single depolarizing pulse, the bacterial sodium channel activates and then inactivates with a time course or a half time of around 8 to 10 milliseconds. So this is about 10-fold slower than fast inactivation of a vertebrate channel, which is in the 0.8 to 1 millisecond time scale. If we repetitively stimulate the bacterial channel, just like the mammalian channels, slow inactivation accumulates, and if you stimulate at a rate of only 1 hertz, which is not very fast, the channel is completely inactivated in 10 seconds and very hard to recover from that state. So slow inactivation is a very active process in NAVAB, and we think it's the most stable state of the channel at 0 millivolts, because when you get to here, it's very hard to get the channel back. So here, I'm showing part of the structure of NAVAB wild type. Remember previously we were looking at a single residue mutant. We were fortunate to get the structure of NAVAB wild type, and we believe it's in the slow inactivated state, as you would predict from this physiology, and you can see that on the right here. Now the selectivity filter, instead of having a square array of glutamates, is distorted and has a parallelogram of glutamate side chains. In the center, in the central cavity, instead of having a square array of the relevant side chains here in colors, you can see that there's a parallelogram array. And at the intracellular end, instead of having a symmetric distribution of the 4 S6 segments around the activation gate, 2 S6 segments have moved toward the central axis and 2 have moved away, again creating a rectangle or parallelogram structure. So this asymmetric pore collapse, we believe, is the structural basis for slow inactivation of bacterial and mammalian sodium channels. So sodium channels are important drug receptors, and particularly important about once a year for all of us when we go to see our dentist, because it's local anesthetics blocking sodium channels that keeps your brain from knowing the bad things that the dentist is doing in your mouth. And not only local anesthetics, but sodium channel blocking drugs are used in treatment of epilepsy, cardiac arrhythmia, and chronic pain. So they really are very important. So how do those drugs work in a functional and structural sense? So here, the modulated receptor hypothesis is illustrated. This is a hypothesis by my colleague Bertil Hille, formulated in the 1970s, so long before there was any structural information about sodium channels. So this is based entirely on biophysical studies. And so Bertil and other members of the field at that time postulated that local anesthetics bind in the pore. They must reach their binding site from the intracellular side, and they reach their binding site much more easily when the activation gate is open to allow the drug binding to move into the pore. And this confers on these drugs frequency dependence in a tissue in which the sodium channels are open quickly, like a cardiac arrhythmia or an epileptic seizure or a chronic pain in a nerve. These drugs rush in and block much more effectively, and that's why they're therapeutically useful. Also, 2 other points came from this early work that's very important. First, drugs can enter through the open pore, but small hydrophobic drugs can also reach this receptor site through the membrane phase and form resting state block. And the affinity for drug block is higher in inactivated channels based on this early biophysics. And kind of amazingly, all of this has turned out to be true at the structural level, as I'll show you in the next couple of slides. So first, again, starting at the mammalian channel, a model from the era of about 2000, so a model before crystal structure, shows a local anesthetic molecule, lidocaine, in a receptor site formed by 3 transmembrane segments that line the pore. The 1S6, 3s6 and 4s6 transmembrane segments here, lining the pore, and in space-filling representation, the side chains determined by site-directed mutagenesis that are important for drug binding. So if we imagine the life of a local anesthetic drug, so if you're a local anesthetic drug floating around in the cytosol here, and you need to block a sodium channel, then you have a very short time to do so, right? This channel opens, now the drug binds, inactivation gate traps it in place, and drug-bound inactivated channels are very stable, and this is the basis for state-dependent block of local anesthetics and the other related drugs that I mentioned. Now, in terms of real three-dimensional structure, the relevant amino acid residues are located in the positions of the colored balls here, in the local anesthetic binding site, in the central cavity of the channel. If you look carefully, there's a fenestration that leads to this site and would allow access of small hydrophobic drugs. And looking down on the pore, local anesthetic drugs bound in this site would clearly block sodium movement. We think this is the structural basis for block of the pore by local anesthetics. Moreover, as I showed you, this site changes conformation in the slow inactivated state. The key binding residues go from having a square conformation to a parallelogram conformation, and we believe this is the structural basis for high affinity binding of local anesthetics in their site in inactivated sodium channels. Now I have one more topic and two more slides, so I have to ask my chair, can I go for it? Okay, so remember I told you that bacterial sodium channels are the ancestors of mammalian sodium and calcium channels. So we wondered, how can we make a calcium channel out of NAVAB? So a lot of work by other groups had preceded us on this and had shown which amino acid residues are important for calcium selectivity in mammalian calcium channels. So we introduced those into NAVAB. There are 3 molecular changes. So this is the NAVAB selectivity filter, TLESWSM. E is the high field strength site that I have emphasized before. L and T form the central and inner sites. And these residues to the right, or on the C terminus of this high field strength site, are uncharged in NAVAB. They form the outer vestibule, and you can see that illustrated here as a sodium or calcium ion would see it. If we make 3 molecular changes, the most important one is S to D, adding a charge. The second most important one is M to D, adding a second charge. And the third change, not so important, E to D, also helps with calcium selectivity. We change the ratio of calcium to sodium selectivity from 0.03, that is 30-fold sodium selective, to almost 400-fold calcium selective, a 12,000-fold change. And the structural basis for that change is shown here. Calcium ions line up in this crystal structure of our calcium-preferring form. We call it CAVAB with tongue-in-cheek. And you can see 4, actually 5 calcium ion binding sites, 3 in the heart of the selectivity filter here at D178, a new site that we introduced, D177, the high field strength site, T175, the inner site. Here, methionine 181 used to be, now N181. That's an additional site we introduced, and then this is an ion in the vestibule. So calcium ions visit that series of sites as they move through the pore, and if you don't have that ladder of calcium binding sites to allow one calcium ion to knock off the other into the cytosol as they're transported, then you don't have calcium selectivity. In support of that, divalent cations that block calcium channels and CAVAB, such as manganese, and cadmium, bind at the high field strength site, but only one ion binds in the pore, blocks it, and prevents conduction. Okay, so I've stolen a couple of minutes from our coffee break, and now I want to finish up. So that's our story of how sodium and calcium channels work so far. Of course, there's more things going on, and I can tell you some things in conversation that didn't get onto the slides this morning. I won't take time to mention all of the names on this slide, but I do want to say that this is many years of work, so many former graduate students and postdocs played critical roles. These are the people that played key roles in the data that I summarized for you. There are many collaborators, but I want to highlight my structural biology collaborators, without whom none of the main part of the talk would have been possible. Ning Zhang, crystallographer in pharmacology in Seattle. Rachel Clevitt and David Baker in biochemistry. Rachel for NMR analysis and David for molecular modeling. And of course, thanks to NINDS and NHLBI for the essential funding. And thanks to you for your attention.
Running a minute behind, not because of the speaker, but because of the generous introductory sessions. We do have time for a question, I think, in the back.
Just throw it.
Yeah. You should come sit in the front. You wouldn't need a microphone.
I need to see how old I am, Bill. So,
I can't really see who you are. It shows how blind I am.
yeah, it's Mark Ellison.
Hi, Mark.
So, the slow inactivation puzzles me. Myelinated fibers, and for example, the auditory system really can't suffer very much conduction velocity change with activity. I wonder if there are differences among the sodium channels with regard to the characteristics of slow inactivation, and if that would accommodate it.
We believe all sodium channels have slow inactivation, but it takes place at different rates. And one of the interesting spin-offs of our early biochemistry of sodium channels was the discovery that they're substrates for pKa phosphorylation. That's important in Seattle, where Ed Krebs was my predecessor as chair. And that pKa phosphorylation adjusts the slow inactivation process. So, for example, serotonin or dopamine-dependent pKa phosphorylation of sodium channels reduces the rate of slow inactivation in cortical neurons. So we think this is a kind of regulable and adjustable process and is adjusted to the needs of the particular system.
Let's thank our speaker again. So one of the treats of this symposium and one of the treats of modern science is the number of graduate students that Palmer has generated and seeded the intellectual community with. They cover quite a span of years, and we're going to hear from two of them in the rest of the section. Next speaker is Steve Sine, who's a professor at the Mayo Clinic and has been a leader in work on nicotinic receptors. Steve?
And coming through loud and clear, one of Palmer's first students, it's a great honor to be here and to reflect on what has been a tremendous arts accomplishment. And what I'd like to do today is really to connect the dots between the absolutely foundational work I did with Palmer as a graduate student to what I'm doing today. And in doing that, I'm going to break my talk into three parts. Basically, I'm going to start with some of the experiments we did together. And then I'm going to maybe give you a little bit of background to understand the kinetics, which is the body of the talk. And then the body of the talk will be entirely in the spirit of the symposium and in my relationship with Palmer. Completely unpublished data. And so the body of the talk will be very much like a conversation that I would have with Palmer, telling him what am I up to these days. And that's really what I'm going to talk about. I'll begin with the central question. We study the nicotinic receptor found in your nerve motor synapse. This is what governs moment-to-moment communication, allows you to move around on a moment's notice. And light molecules within the brain allow communication between neurons on a moment's notice and are responsible for all of your higher order thinking and responsive and perception. So the central question, I guess I'm supposed to point to this one. Central question, we've got these membrane-embedded receptors. They they respond, they bind nerve release transmitter, and in response to that, open an ion channel, causing this opening and closing of the channel and very rapid flux of ions through the membrane. The central question is, how do these work? And we were approaching that question at a very basic level 30 years ago, and now we're continuing to refine this process in further depth. It's also a serendipitous pleasure to acknowledge Bill Catterall. And the reason I'm acknowledging Bill is I'm starting now with the work that I did with Palmer, and I came into the lab, and Greg Weiland, Shelley Camp, and Brian Georgia were busy doing ligand binding assays on torpedo acetylcholine receptors. And they were relating the results from ligand binding assays to functional state of the receptor. But we didn't have a measure of functional state of the receptor at that time. And so Palmer, through his encouragement to develop my own independent thinking and to do whatever I felt like doing, and in particular said, do what matters for the problem, not what everyone else is doing. And so going through the literature, I noticed that Bill had worked out a very convenient assay to monitor function of sodium channels as well as muscle acetylcholine receptor channels using isotopic sodium radiotracer uptake. And so this was a true inspiration. Thank you, Bill. And this is one of my first experiments. What we're doing here is measuring sodium uptake by cultured muscle cells in response to carbamylcholine added to the cells. You can see there's very rapid uptake, and it's more rapid if the cells are not incubated with the agonist first. The second trace is with the agonist pre-incubated. And it's a highly specific flux, because this is the flux that you get when there's bungarotoxin pre-bound to the cells. So we had a measure of response, and we had measures, of course, ongoing in the lab, measures of ligand binding. And so in one of the papers, I'll just highlight this one paper that we did together, we looked at the relationship between occupation by agonist and the permeability response revealed by bound cobratoxin. And the experiment went something like this. We bound particular different fractions of the receptor with bungarotoxin, or actually cobratoxin, and then in the same cells measured the response. How much was the response reduced? And here are various models for this decrement in the response. And here are the actual measurements. And I was particularly proud of these at the time because these are double isotope measurements, because the bungarotoxin was labeled. It gave a direct measure of the occupancy, and so was the sodium label, and they could be counted separately in the gamma counting on different channels. And so it's not a linear response. This is one of, I think, 7 figures in that paper. They were all curvilinear inward, and they were best described by a model in which there were 2 binding sites, and the only way the receptor would function is if they were both free to bind the agonist. And so this is a concept that prevails even today in what you're going to hear about. Second experiment we did was to, instead of looking at function, we looked at the ability of the agonist to compete against cobratoxin, a measure of binding. And what you'll notice is the curves, binding isotherms are steep with no occupancy. This is without any bound toxin. But with 85% of the sites bound with toxin, the remaining sites bind anti-cooperatively. And we interpreted that as a result of different agonist affinities of the 2 sites. And this is encapsulated in a model from our paper where the top plane is the activatable receptor, the bottom plane is the desensitized receptor, and then we have receptors with 2 binding sites, A and B, that are distinct in their affinities for the agonist. The only species that can activate is the doubly bound species. And so these were 2 conclusions from that work that prevail even today. And so I'm going to now go into the second technical part of the talk. It'll be just about 5 minutes. I'm going to tell you what we do on a daily basis, and this will enable you to understand the detailed kinetics I'll go through. And so what we do on a daily basis is we have these HEK293 cells. We transfect them with receptor DNAs. The receptors are on the surface within a day. And we come in with a patch clamp electrode, something I learned in my 2 postdoctoral fellowships, make a gigaseal, and the electrode's loaded with agonist. And with a membrane potential applied, this is what we get. This is current as a function of time. This is a single ion channel opening and closing and then at the end, closing altogether. So what do we do with this kind of data? We do what we call dwell-time analysis, where using a computer, we idealize the events. We take a look at these events, measure the open times and the close times, and then we put them into logarithmic histograms like this. And this is just 20 events. We do openings and closings. If we do tens of thousands of events, we get these bell-shaped curves, which on a log scale reflects a single exponential. And if we fit them with a kinetic scheme, this here is illustrated as the simplest kinetic scheme. We have the open channel and the closed channel, each contributing an exponential component. The importance of this is from the mean open time and mean closed time, we get a measure of the transition rates between the two states, open and closed. And you'll see histograms that are much more complicated than this. They'll have more than one exponential apiece. But the point is we get an idea of the energy landscape of the protein from these kinds of measurements. And so I'd like to introduce Nargis on the left and Corey on the right. They did all of the recordings I'm going to show you. Nargis is a postdoc, and Corey just got his first faculty position and was a postdoc for this work. And so the work I'm going to tell you about was really motivated by two papers that came out in Nature about 9 months apart, about 8 or 9 years ago, not quite that far ago, 7 years ago. And the essence of both papers, one from Lucia Civilati and one from my lab, was the detection of what we call priming intermediates. These are closed state intermediates between the resting closed state and the active open state. These are significant because these are the states that the receptor must go through in order to get to the open state. They're relevant for things like partial agonism and for the functioning activation process of the receptor. And what I want to show you now is a trace, a typical trace from these experiments. This is at what we call 10 kilohertz bandwidth. I want to point out one of the limitations in that work, and that was most of the evidence for these intermediate states came from these partially resolved closing events. These are the intermediates that we're barely able to see at 10 kilohertz bandwidth. And so the simple solution is, well, increase the bandwidth. The patch plant can actually go to a much wider bandwidth than 10 kilohertz, but we were facing a signal-to-noise problem. And so if you look at this, 25 kilohertz, it's detecting these events, these openings and closings better, but we're getting lots of false events. So in order to get good detection of events, we needed to have the events bigger. So we did that through some trial and error. We found a special patch solution that would tolerate very high voltages, and we made our channels bigger by increasing the voltage. Now you can see 10 kilohertz is plenty of bandwidth, and 25 kilohertz we can go and detect the events without spurious false events. So what I'm going to tell you about is we've got this new tool, and this is what we've done with it. Consequences of this are, if we make, say, a closed duration histogram from these data, at 10 kilohertz, this is what it looks like, and this is fitted by the sum of exponential components. What I want you to notice is the briefest component is barely resolved. One wouldn't want to rely on data like this, but this is what we relied on for evidence for these intermediate events. So what happens with higher bandwidth, we get about half a decade more bandwidth. So the shaded boxes represent the extra bandwidth we get at 25 kilohertz. I want to point out that the number of events detected, say, briefer than 100 microsecond is more than double. Notice the square root scale here. So it's actually much, much more advanced. So this is a typical experiment. This is with acetylcholine as the agonist. This is a cluster of events from one single receptor. We analyze the openings and closings to get insight into the energy landscape. These are the closed duration histograms, the open duration histogram. And what you're seeing here, the smooth curves are probability density functions obtained by kinetic fitting of a mechanism to the data. And I'm going to just show you, furthermore, what we do is not just one concentration, but a range of concentrations, so we can sample a condition where most of the receptors are in the inactive state, and then they go the opposite condition where most of them are in the open state. This allows us to eliminate lots of possible mechanisms that could possibly describe the data at one concentration. So this is the mechanism. This is what we call the prime mechanism. It has three dimensions to it. There's a binding dimension, binding of neurotransmitter, a priming dimension, and then a gating dimension. I could spend a few minutes on why we selected this mechanism, but suffice it to say that this is the simplest mechanism that can describe the data. And what I'm going to do now is tell you how each of these steps depend on the extent of occupancy, the efficacy of the agonist, and a gain of function mutant from a disease situation. So first of all, before I get to that, I'm going to tell you, because we're on the very edge of detection and we're in uncharted territory, we thought it was essential to support our results, our experimental results in fitting using simulation. So what I'm showing you are some stochastic data sampled at 500 kilohertz, filtered at 25 kilohertz, and then with added computer modeled noise to mimic the open and closed channel noise. What I want to show you is that we're able to actually, if we look at doing all points histogram, that is plot the amplitude of each of these digital points here. This is the baseline current, this is experimental, this is the open channel current. The filled in area is the simulated data, and the outline is our experimental data. So these agree quite well and further support our fitting analysis. So, and these are what the simulated data look like, and just to put them side by side, the experimental data are right here. And so we have, again, sets of dwell time histograms, single channel traces, and by inspection you can see there's good agreement. Furthermore, the rate constants gotten by fitting the same scheme to both sets of data are within about 10% of one another. So I'm going to talk now first about the binding. From now on, it'll be just what were the rate constants that we got, and we're going to associate them with occupancy. Okay, so the first thing are the binding steps, the association steps. We've got good values for those. Binding to the resting state turns out to be, didn't go right away. So about 10 to the 8th per mole per second, identical values for both binding steps. And this is about 10-fold slower than the theoretical rate of diffusion. Okay, and so from this we can conclude that the association is occupancy dependent. And association to the primed state, to our surprise, was about 10 times faster. And so association is also state dependent. And looking at the agonist association rate constants, we see that we get very different association rate constants for the two binding sites, and so they are occupancy dependent. But the dissociation from the prime state is pretty similar to that from the resting state, so I'll say that's essentially state independent. Okay. And so these produce the following agonist KDs that turn out to be occupancy dependent and state dependent, but for different reasons. One depends on the association rate, the other on the dissociation rate being different, depending on state or occupancy. So now I'm going to consider the priming rate constants right here. For acetylcholine, this is the first priming step. It doesn't go very well. It's about 1%. The second one, it goes much better. And these are the rate constants. But what was really interesting is, it was occupancy dependent, but what was really interesting was to look at the priming rates versus the equilibria, comparing the forward rates for priming with the equilibrium constants for priming. The ratio of the rates was 60, but that for the equilibria was only 20. And so, and furthermore, the change in the log of the rates versus the change in the log of the equilibrium constant was 1.3, and I'll touch on this in a second. The main point here is that occupancy enhances the rates more than the equilibria. And I'm going to tell you more about this for a partial agonist and also in contrast to the gating reaction, which I'm going to tell you about next. So the gating step, right here, you've had singly ligated opening and doubly ligated opening. And first of all, I'll tell you from the rate constants that this is occupancy dependent. Gating is not very good with a single occupancy and is much better with a double occupancy. And these are very fast rate constants, so it really did require our bandwidth improvement to see them. And again, looking at the gating rates versus the equilibria, the ratio of the forward rates, 9,000 to 130,000, compared to the equilibria, about 170 is the ratio. And so we see, again, that we look at the change in the log of the rates versus the change in the log of the equilibrium constant. This is much lower, between 0 and 1. But what we conclude from this is that occupancy enhances the equilibria more than the rates. And this is just the opposite of what happens with the priming step. And this is significant from the viewpoint of rate equilibrium free energy relationships, where a number between 0 and 1 implies that the transition state is a hybrid of the two flanking ground states. When this is greater than 1, we conclude that that this process involves a stabilization of the transition state. So, okay, so carbamylcholine, turns out this was the agonist I used for most of my thesis work. We thought it was a full agonist, but I'm going to show you that it is a partial agonist. This is carbamylcholine here. These are single channel traces and 12-time histograms. Even the highest concentration, you can see the channels don't open nearly as well as with acetylcholine. And this is why I call it a partial agonist. Well, we did the same analysis that I just described to you with carbamylcholine. And I'm going to emphasize just the priming and the gating reactions. There's also an effect on the association steps, but I'm going to skip that. So the priming steps, you can see with carbamylcholine, singly liganded priming is not very fast, it's not very efficient. That for doubly occupied priming is better, but not nearly as good as with acetylcholine. And this is why it's a partial agonist, in fact. So we look at the ratio of the rates. This ratio is about 20. And the ratio of the equilibrium constant is about 21. So in contrast to acetylcholine, they're essentially the same. So if you take the ratio of the change of log of the rates to the log of the equilibrium constants, you get a value just under 1. So it's within this range of linear rate free energy relationships. And the main point is occupancy with a partial agonist enhances the rates and the equilibria to an equivalent amount. And this is a surprise to us. When we started out with this, trying to enhance our bandwidth, we thought we were doing a technique, but all of this insight into partial agonism came forth out of the measured rate constants. And gating, let's take a look at singly liganded and doubly liganded gating for carbamylcholine. You see the rates ratio, equilibria ratio, and again, this is between 0 and 1, much as we concluded for acetylcholine, where it falls into this linear rate equilibrium free energy relationship. So the conclusion from this is occupancy enhances the gating equilibria more than the rates, just as we noticed with acetylcholine in the wild type receptor. So the final aspect that we applied our higher resolution recordings to is this mutation discovered by Andy Engel, my colleague at Mayo Clinic. This is in a person with slow channel myasthenic syndrome, a devastating muscle weakness disorder. And I'm going to just show you what the analysis looks for these channels. Here's acetylcholine. You can see the channels open much better with this mutant, and to show you even better, here's the wild type receptor right here, and to give you an indication, here is 10 micromolar acetylcholine. Right here, you can see the openings are very, very much biased towards the open state. The same concentration with the wild type receptor is much less. So the problem with this receptor is it's activating too well in this patient. That's why the patient is sick. Okay, so what about the analysis? What does it tell us? Well, I'm skipping the part where the mutation enhances the association and slows the dissociation rates. It does affect affinity, but I'm going to focus on priming and gating again. Well, if you look at the forward rates, their ratio is 66, and the backward rate in the equilibrium is only 4. So this delta log rates over delta log equilibrium constant, way far away from 1, and it's 3.1. And so the conclusion here is that the occupancy enhances the rates much more than the equilibrium. So this essentially makes acetylcholine a super agonist, one that you never find in nature, other than in this patient. Gating is a similar story to carbamylcholine and acetylcholine on the wild type receptor. We compare the ratio of the rates to the ratio of the equilibria. The log of the rates to the log of the equilibria is between 0 and 1. Again, the transition state is a hybrid of the flanking ground states. And again, occupancy enhances the equilibria more than the rates. So this is really a disease of the priming step, and also, I didn't talk about it, but also of the association-dissociation steps. Okay, I'm going to just summarize. Here's the scheme. We've got binding, priming, and gating. And we'll summarize the binding first. We think about occupancy dependence and state dependence. The association rate constants highlighted in color, they are not occupancy dependent. However, they are state dependent. The dissociation rate constants, highlighted again in color, are occupancy dependent, but they are not state dependent. So we see this opposing juxtaposition of the on race and the off race from this analysis. So the next dimension is the priming dimension, again highlighted by the colored arrows. Priming is, as I told you, is occupancy dependent. The rates are more dependent than the equilibria on occupancy, and this is true for acetylcholine. However, this does depend on efficacy. If it's a lower efficacy agonist, this dependence is about equivalent. And however, for a gain-of-function mutation, this dependence is even more exaggerated. The gating was the common thread through all of this. It doesn't change too much. Occupancy dependent, however. The equilibria are more occupancy dependent than the rates by a long shot, and this falls into the linear rate free energy relationship situation. And however, this relationship is independent of the efficacy, the same for a full agonist and a partial agonist, and it's also independent of a gain-of-function mutation. So I'd just like to tell you, well, what might this be important for? I think as many of you may know, that the muscle receptor, which is the topic of the talk today, is part of a big family, a super family of pentameric ligand-gated ion channels. And I think the lessons we glean from this muscle receptor, which is now can now be interrogated at very high resolution, will play out in other members of the family. In particular, receptors activated by glycine, GABA, serotonin, and of course the neuronal nicotinic receptors in particular. You're going to hear about them in the next talk, this receptor that's associated with smoking addiction. The other relevance is for diseases of the synapse. This is a cross-section through a nerve muscle junction from a normal person. Notice the close juxtaposition between the nerve ending and the muscle. This is the same image from a slow channel syndrome. I'm not sure if it was the one that I showed you, but what you can appreciate is the nerve is starting to pull away from the muscle, so that even though it's releasing transmitter, it's not getting there very well. And in a sense, the body wants to get rid of this nerve because it's overstimulating the muscle. So this is the response. Well, and this is a model of the receptor, and what I've done is just highlighted mutants that over the years Andy Engel and I have characterized kinetically. What I've shown here, the locations in green are mutations that cause slow channel syndrome, or one where the response is too big, like the one I showed you. Those in red are sites where mutations cause a lower response than normal, and they're called a fast channel syndrome because typically the responses are small and very brief. And I think since we've characterized these with low resolution, we've made some preliminary conclusions about their mechanisms of action. With this better resolution, we're now in a position to distinguish whether the mutation alters the binding steps, the priming steps, or the gating steps. And I will close. Thank you for your attention.
We certainly have time for few questions. Jean-Pierre.
In your dynamic schemes that you show, the open state is always a bound state. Okay. Now it is well known that there is spontaneous activity without ligand, and also that many mutations, which are called constitutive mutations, do open the channel without having the ligand bound. So why did you omit them?
Well, because we can't detect unliganded openings in the wild-type receptor. Also, in this mutant receptor, you put a patch electrode on the cell, you don't see anything. So I want to emphasize that the scheme that we have is the simplest scheme that can describe the data. If we don't see transitions, we can't put a state in. And
This is seen in many other receptors of the family.
yeah, absolutely. This is just happening at too low of a frequency to be able to detect or to associate with the rate constant.
Palmer.
So, as I understand this, Steve, and I'm going to have to go over it a lot, but what you're saying is that the absolute rate theory of IRE, where that transition state is towards occupancy or not. So then one of the interesting questions would be how you get this priming, because the two alpha subunits are not together, but they got to communicate in some way in that pentamer.
So what was a surprise to us was to find out that the rate constants for the priming step fell out of this linear rate free energy relationship. Actually, if you look at some of the literature, there are examples from some enzymes where they plot their rate versus the equilibrium constant as a straight line with a slope of infinity. So they, I think it was, I forgot the name, but anyway, the interpretation was that these selectively affected the transition state as opposed to the priming states, as opposed to the ground states. And so what I think is happening with these full agonists and with this disease-causing mutation is we're not excluding effects on the ground states, but the effect on the transition state is more than we can account for by just effect on the ground states. So at first I didn't believe it until we saw this example from all of these different partial agonist, full agonist mutation. There's a couple other examples where the priming steps and the transition rates are changing in this way, but the gating steps are all the same. They all give this ratio between 0 and 1. So this is hot off the press. I welcome a discussion and possible interpretation. But I think the take home is that for the priming step, the occupancy affects the rates much more than the transition, or much more than the equilibrium constant for gating or for priming.
I think we better go ahead and move on, but thanks, Steve. It's a very nice presentation. And our final speaker before the coffee break is another former graduate student of Palmer's, Ryan Hibbs, who's now an assistant professor in biophysics and neuroscience at UT Southwestern. So it's getting to the era of graduate students that I had something to do with since I joined the faculty.
All right, so it's a tremendous honor to be here. Thank you very much for the invitation. I have to say I'm pretty intimidated by the audience and the panel speakers that I'm lucky enough to be a part of. So to give you a sense of how Palmer's influenced my career, I wanted to talk about where I came from, what I started working on in Palmer's lab a little bit before we get into the recent results. So I started graduate school in 2001 here at UCSD, and I was a member of the receptor group, just like Steve, but about 20 years later. And a little bit changed in that time period. So right when I joined in 2001, the first high-resolution structural information had come out on superfamily of pentameric receptors, and that was in the form of the acetylcholine binding protein, which we heard a little bit about last night. And it's analogous to the extracellular domain of the nicotinic receptor, so it's this sort of upper portion, and it forms the bindings, has the binding sites for ligands. And that really propelled the forward field and allowed us to ask a new kind of level of detailed questions about mechanism in these receptors. And so I started taking this new crystal structure, this acetylcholine binding protein, and made a large panel of cysteine mutants all over the surface of this protein, labeled those engineered cysteine mutants with fluorophores, and probed the effect of ligands on conformational changes in the receptor, and used the change in fluorescence to map ligand binding sites. And while this was going on, these solution-based structural studies of this receptor that we now have the structure for, I was watching Scott Hansen in the lab, who was a graduate student at the same time. He started 1 year before me. And he saw this first structure and decided he needed to get new structures of this protein with different ligands bound. So I watched him trying to learn crystallography, and he worked with Susan Taylor's lab and Gori Ghosh's lab here and, at the beginning. And he was getting some crystals to grow, and they were diffracting poorly, and I was watching him struggle through building models into low-resolution electron density maps. And I said to him and other people, I would never do crystallography. This is just terrible, too risky and just painful. But then Palmer got behind this full bore and sent Scott to France, where he was able to finish up a really beautiful set of these structures that I doubt we'll talk about today, but really illuminated some initial principles of allosteric gating in these receptors, local conformational changes around the neurotransmitter binding site. And I saw this and all this detailed information you could get from crystallography, and I was convinced then that this was a worthwhile thing to learn. So Palmer sent me to France as well, and I learned how to do structural biology with Eve and Pascal over there, and we got some nice structures with partial agonists bound. And at the same time, my wife came over, Colleen, who also did her PhD here at UCSD, and joined me at the end of my time in France. And I proposed to her at the end of that trip, and she said yes. And I think that being in southern France was no doubt integral in getting her to accept my proposal. So Palmer's influenced me in many ways, professionally, personally. Okay, so we have these structures of binding proteins, these solution-based studies of structural changes. And it became clear to me then that we were limited by the lack of structural information for intact receptors. And so that's what I went to do in my postdoc work, and eventually was successful in getting a structure of a eukaryotic cis-loop receptor, a pentameric receptor, the glutamate-gated chloride channel from C. elegans, which has been in the news recently because it's the target of ivermectin, which was related to the recent Nobel Prize. So that was one structure, and that helped me get my faculty position. And then what I really wanted to focus on was nicotinic receptors, going back to what I first became interested in working with Palmer, because we still lack high-resolution structures for nicotinic receptors. And I do not have a high-resolution structure for a nicotinic receptor to present to you today, but we're on our way. And I'm going to tell you today about the methods we're developing to work with these receptors. Okay. All right, so we're doing crystallography, and so these are, crystals of nicotinic receptors. We're also doing cryo-EM, and these are 2D class averages. And then that's just a pretty picture, right? Okay, all right. There we go. All right, so I want to place nicotinic receptors in the larger context of the superfamily they belong to, called cis-loop receptors, or pentameric ligand-gated ion channels. Steve touched on this superfamily at the very end of his talk. So we have this superfamily can be broken into two broad categories: those whose ion channels are selective for cations, and that would include the nicotinic acetylcholine receptors and 5-HT3 serotonin receptors. Then we have anion selective receptors like the GABA-A receptor and the glycine receptor. These cation selective receptors, exemplified by the nicotinic receptor, typically play an excitatory role in synaptic transmission or nerve muscle transmission. Anion selective receptors typically play an inhibitory role in synaptic transmission. All right, regardless, all of these receptors share a conserved architecture. They're all pentameric. They can be homopentameric or heteropentameric. The vast majority in humans are heteropentameric. Each subunit in the pentamer also shares a conserved architecture. They have a large extracellular ligand binding domain at the N-terminus, and 4 transmembrane spans. And there's a large intracellular loop that's poorly conserved among the different family members between the 3rd and 4th transmembrane alpha helices, and I'll come back to that loop in a couple of slides. Okay. So what are we interested in understanding about these receptors? The classical things that Palmer talked about a lot. So principles of ligand recognition. where do these different classes of small molecules bind? How is the interaction of antagonists different from agonists? Where do positive allosteric modulators bind? Principles of ion permeation, so why are these cation selective receptors? Why do they only let cations through? Why do some of the cation selective receptors let a lot of calcium through, and some are essentially impermeable to calcium, etc. Allosteric activation, so this is a huge deal in the field. Everyone wants to understand how binding of something like acetylcholine, 50 or 60 angstroms from the gate in the middle of the membrane, opens that gate, stabilizes an open conformation. And there are a lot of ideas out there, and we're starting to get our heads around a structural basis for this, but I think it's fair to say it's not that well understood yet. Okay. But we have learned a lot about all of these things from classical pharmacology, biochemistry, and more recently structural biology, and we'll hear about some of this, I think, from Pierre-Jean Corringer this afternoon about gating. He's done some beautiful work recently. All of the structural information that's come out, though, has been from homopentameric receptors. And we have now structures at pretty high resolution for the serotonin receptor, the homopentameric GABA-A receptor, and a glycine receptor. We have no high resolution structures of heteromeric receptors, and I think that that is going to be the next major milestone in the field, major hurdle, allowing us to understand at a structural level the relationship between subunit stoichiometry and function. And I want to take one step a little bit even broader and point out that this issue of heteromeric receptors is not unique to this pentameric receptor family. Trimeric, most families of channels, oligomeric channels and receptors are abundantly populated by heteromeric members. So the trimeric receptors, the tetrameric channels like potassium channels and glutamate receptors, as well as the pentameric cyclic receptors and hexameric gap junction channels are all abundantly populated by heteromeric members. And getting structures of these is a major challenge. And so that's what we want to go after. Okay, so the target and what I'm going to talk about today is the alpha-4 beta-2 nicotinic receptor. So this alpha-4 beta-2 receptor is the most abundant nicotinic receptor in the brain. And it is not only an obligate heteromer, but it can assemble in two functionally distinct stoichiometries. 3 alpha plus 2 beta here on the left, and 2 alpha plus 3 beta on the right. Both of these receptors are made, they're found in the human brain, and they have different functional properties. So the one on the left has a higher calcium permeability than the one on the right. The one on the right has about 100-fold higher affinity for acetylcholine and nicotine, and the one on the right, 3 beta 2 alpha, is selectively upregulated by nicotine in systems ranging from cultured cells to smokers' brains. And this is the key receptor in nicotine addiction, as Jean-Jeu beautifully showed with knockout mice, that if you take these subunits out of rodents, they will no longer self-administer nicotine. So there's great physiological implications. Both these stoichiometries are functionally important. Making one of these receptors recombinantly and somehow getting a high-resolution structure for it is going to require somehow making or purifying selectively one of these stoichiometries. And so that's really what this talk is about today. So the outline for the talk, working toward a high-resolution structure. I'm going to start by telling you about some small-scale expression tests we did at the human receptor, go through how we figured out how to scale up to milligram production scale, present a new biochemical assay for stoichiometry based on fluorescence, and then move into crystallization of this receptor. All right. Okay, so on the left, I have another version of our cartoon here showing the subunit topology for a nicotinic receptor or any pentameric ligated ion channel. We have the extracellular domain that makes the neurotransmitter binding site, the 4 transmembrane spans, and an M3, M4 loop. And I highlight this loop because this is where we are putting GFP for the purposes of small-scale expression tests. So what do? We do something we call fluorescence detection size exclusion chromatography, or FSEC. And this is just like simple gel filtration or size exclusion chromatography, but we instead of monitoring absorbance like one typically does, we monitor GFP fluorescence. What this allows us to do is to look at a gel filtration profile of our protein without having to purify it. We can look at it in the background of whole cell extracts. We need nanograms to micrograms instead of milligrams to do one of these experiments. So as an example, this is a positive control. Here we're looking at GFP fluorescence versus time coming off the gel filtration column. And the example trace I'm putting up here is GluCl, or this glutamate beta chloride channel, which I mentioned we crystallized earlier, and is our standard for a crystallizable eukaryotic pentameric receptor. So you see one predominant species, and that is the elution volume corresponding to a pentameric receptor. Okay. So we took our alpha-4 and beta-2 subunits, put GFP into the intracellular loops, transfected the two subunits into HEK cells, solubilized the whole cells, ran the extract over a gel filtration column, monitored GFP fluorescence, and crossed our fingers. And much to our delight, this was a very promising result. We saw that the predominant species came off at the elution volume we expected for a pentameric receptor, and it was a fairly tight eluting peak. So that suggested this was worth pursuing. All right, so we then went through a battery of mutants where we tried to truncate the construct to make it as a minimal functional construct for the purposes of structural study. And most of those deletions were in the area of the linkers that connect the GFP to the receptor. And we came up with our truncated construct, did the same thing, GFP is still there, and we see an increase in expression, even better than GluCl, a narrower peak, and a shift to the right corresponding to a smaller, lower molecular weight, which is consistent with the fact that we made some deletions in all 5 subunits that are in this pentamer. Okay, then we did a control experiment where we just transfected the subunits alone and showed that they do not substantially contribute to forming homopentamers by themselves. All right. Okay. So what were the next hurdles? So I just showed you that we had these small-scale expression tests that suggested great biochemical behavior, reasonable expression level. So what was next? We had to scale up. So our go-to system for scaling up to the milligram production scale of protein was insect cells. So we made baculovirus for these receptor constructs and infected insect cells with them. And the protein just looked terrible, not a predominant pentameric species. So we needed to step back. And others had recently been coming out with high-resolution structures of proteins made in mammalian cells using so-called BACMAM virus, which is baculovirus, just like you make in insect cells and for infecting insect cells, but instead in the expression vector, you replace the insect cell promoter with a mammalian promoter, like a strong CMV promoter. And then you can take this baculovirus, dump it on HEK cells, and it will transduce expression in HEK cells. Terrific. So we made some BACMAM virus, figured out how to culture these cells in suspension, and we saw some nice behavior, but the expression level was very low. And we also saw that as we added more and more virus, we got more and more protein, but we were limited by viral titer. Okay. So we needed a way to get higher titer virus, and we needed a way to titer our virus more quickly. Classical titration assays for virus take plaque assays, take a week or 2 or 3. We're looking at hundreds of constructs. That's not going to work. Another justification for coming up with a rapid titering assay for this virus was we had this mixed heteromer issue. We needed to be able to control the ratio of virus for subunits that we were adding in a rigorous way. Okay, so to get around these problems of low expression levels in the BACMAM system and this mixed heteromer issue, we started with a new expression vector. And we call this vector PEZT for easy titer plasmid BM BACMAM vector. And this is a slight variation on a commercially available vector. In hindsight, this addition that we made seems kind of obvious, but it's been incredibly helpful to us. So what we have in this vector is we have a dual promoter system. We have a CMV promoter, which drives expression in mammalian cells. And so we put our alpha-4 or beta-2 subunits in this multiple cloning site after the CMV promoter. In the opposite direction, we have the insect P10 promoter, which drives expression of GFP. That allows us, while we're making virus, to follow production of virus in real time. Okay, so we can then do several things with this vector. We can use it for typical transient transfection of HEK cells and our small-scale screening. We can use it for the back-to-back method of making baculovirus that has these TN7 recombination sites. Back-to-back method of making baculovirus is the cheapest, fastest, most commonly used procedure for making baculovirus. We can then use this baculovirus that we make for transduction of protein in mammalian cells, and we can then use this GFP signal for titration of the virus in insect cells. All right, so we made some virus for our alpha-4 and beta-2 subunits. using this system and transduced HEK cells with this virus on a small scale, just a 1 mil suspension culture. And here's what we saw. So we see GFP fluorescence versus time coming off our gel filtration column, and we see robust expression of our truncated construct. And I can't explain the difference that we see here between the truncated construct and the wild type. It's much more profound than we saw before from transient transfections, but it's going in a positive direction for us, so we didn't worry about it too much. Very importantly, these results are consistent. When you're working without titered virus, which is, I mention this because it's common when you're using baculovirus expression in insect cells, people often can get away without titering their virus because in insect cells there's a spreading infection with baculovirus. In mammalian cells, the virus doesn't replicate. So if you don't have a reliable titer or consistent titers that you're using, you get your expression levels are all over the place. So when you're using backman virus in mammalian cells, accurate titers become very important. And we see high reproducibility, which is super important. Okay. So we can then do a more systematic optimization of expression, and here I'm showing you a bar graph of fluorescence from our GFP signal at our pentamer elution volume, and we're just playing around with relative viral MOIs for the two subunits in a systematic fashion. So when we use equal amounts of the alpha-4 and beta-2 virus, we see this expression level. If we start adding some more of the beta virus, increasing the ratio of beta virus to alpha virus, maybe it goes up a little, or maybe that's just noise, and as we add more, nothing else happens. If we shift our playing around with ratio in the opposite direction, we have more alpha relative to beta, we see the expression drop. Okay. When we use this MOI of virus, we actually end up consuming a lot of virus. We're spending a lot of money and time making this virus. And so we wanted to see if we could get away with using less virus. So we dropped the amount of virus tenfold we were using. And of course, our expression goes down. But we have other tools at our disposal. And we also saw then that when we had lower overall amounts of virus, the ratio of multiplicity of infection of the virus became, the expression level became much more sensitive to it. So as we then add more beta virus, our expression goes up quite a bit. We can then add chemical chaperones during expression, including nicotine and sodium butyrate. So nicotine is well known to upregulate expression of this receptor, and as I mentioned before, in a stoichiometrically selective way. Nicotine is polar and uncharged at physiological pH, so it passes through the cell membrane readily. And the mechanism of upregulation is thought to occur at the level of nascent receptors being formed in the ER, and it selectively stabilizes those receptors that have a high affinity for the nicotine. Sodium butyrate is a histone deacetylase inhibitor that acts globally to upregulate expression of everything. And so we can then put these two chemical chaperones together and get a large increase in expression level. And we're now in a zone where we can expect milligrams per liter from cell culture, which is great. Okay, so we figured out how to make bucket loads of the protein. I still haven't showed you purification, but we're still working out these conditions for large-scale expression. So we figured out how to make it, but now we're worried about stoichiometry. We want to get away as much as we can, move toward having a stoichiometrically pure preparation. And we didn't have a great method to do this, a biochemical assay for pentameric receptors. So what we came up with was a dual fluorescent tagging approach. So we put GFP on the alpha-4 subunit, and in the same position, in the beta-2 subunit, we replaced GFP with M-cherry. Okay. So for this assay, we wanted something that would not be tremendously laborious, that would not require many purifications. So we came up with one that requires one purification to standardize instrumentation, after which everything can be done on a small scale. So we do one purification here. We transduce HEK cells on a 1 liter scale with our alpha-4 GFP, beta-2, and cherry viruses. We purify it against an affinity tag. We measure absorbance of the two chromophores. So we measure absorbance of the GFP chromophore and absorbance of the M-cherry chromophore. From the absorbance values using Beer's law, we can calculate the molarity of each subunit. From that, we can get the molar ratio. And in this experiment, we transduced with a 1-to-1 ratio. We weren't concerned about a mixed population for the purposes of this one experiment. And we found out that we did have a mixed population here, no surprise. What we can then do, though, is we can run FSEC with this sample where we know what the molarities of the two subunits are. And we can then correlate the, come back. Okay. We can correlate the molarity with fluorescence values in FSEC for GFP fluorescence and M-cherry fluorescence. And once we have these scale factors allowing us to correlate fluorescence values in our HPLC with our fluorescence detector with the molarities of the subunits, we can then do many small-scale experiments in parallel where we perturb culture conditions and see what the effects are and changes in our estimated stoichiometry. So I'll show you a subset of the results here. So we're looking on the y-axis at the ratio of subunits, alpha 4 to beta 2. So if we had calculated using this scale factor, so if we have a perfect 3 alpha to 2 beta ratio, it'll come in at 1.5, and if it's the reciprocal, we'll be at the 2 alpha 3 beta ratio of 0.6666. Okay, so when we use a 1-to-1 ratio of virus, we have a mixed population that's closer to the 2 alpha to 3 beta ratio. If we use more alpha virus, 2-to-1, 3-to-1, 5-to-1, 10-to-1, we can push it toward the 3 alpha to 2 beta ratio, but it doesn't go there very willingly. If we shift in the opposite direction, we add more beta to alpha virus, we can pretty quickly get down to 2 alpha to 3 beta, and we can push it toward that apparent limit with nicotine in culture. All right. So we use these conditions, this ratio of virus as well as nicotine, with the absolute MOIs I showed you before when we were just going for optimizing expression level, to scale up into large-scale culture for the purposes of purification and crystallization. All right. So here's the results of the purification, and I'm presenting this as FSEC, monitoring tryptophan fluorescence. So here we're looking at not GFP, not M-cherry. In fact, we've taken the two chromophores, the GFP and M-cherry, out of the alpha-4 and beta-2 subunits and replaced them with a crystallization chaperone called Brill. Ray Stevens had a paper on GPCR crystallization a few years ago where he went through a series of thermostable small proteins that have their N and C termini close together and came up with this protein Brill that they found is at least as good, if not better in some cases, than lysozyme in promoting crystallization of GPCRs. So we replaced the GFP and M-cherry with this Brill protein in the exact same place, and this helped us recover some stability as if we had taken, that we'd lost when we took GFP and M-cherry out, and kept our expression levels high. And we hope it would also promote crystallization. So what do we see here? We see a stable protein. It elutes at this pentameric volume as a single peak. We get 2 to 3 milligrams per liter of culture, which is heroic levels for a eukaryotic membrane protein. And it's stable over the course of at least a week in the fridge as a pentamer. So that's terrific. If we run this in STS page, it's not showing up very well, but we have very pure protein, and we know which subunit is which here by tryptic digest and mass spec fingerprinting. Okay, so we take this protein, we concentrate it, we set up broad crystallization screens, and we find that this protein crystallizes readily. So we get crystals growing in the presence of different ligands. These are crystals grown in the presence of dihydrobeta erythroidin, a competitive antagonist. Crystals growing in the presence of nicotine. And to date, our best crystals grow in the presence, best diffracting crystals grow in the presence of nicotine, and I'll show you more from crystals frozen from this drop. All right. So here's some example of diffraction data. This is a crystal in a loop fished out of the drop at the synchrotron. Here's some representative diffraction going to 8 angstroms or a little bit beyond. We now have data that from this crystal form that go to about 6 angstroms. So we're definitely not there yet. We need to get down to 4 or better to answer the questions we want to answer. But it's nice progress. So we can, with the 6 angstrom data set, we can do molecular replacement with different models that are available, then look at electron density maps. And these are certainly biased by the models we use for molecular replacement, but they're fun to look at anyway. So here we're looking down the long axis of the channel, and if you stare at these a lot like we do, you can start to appreciate 5-fold pseudosymmetry that pops out here: 1 subunit, 2 subunits, 3 subunits, 4, and 5. And we'll put the model we use for molecular replacement that we're trying to do some building with now on the right for reference. Here's the pentamer looking down the same orientation. Okay. And if we turn this on its side, here we're looking through the plane of the membrane. We can see low resolution electron density for the transmembrane alpha helices that are just elongated sausage-shaped densities. And then up top we have the extracellular domain, which is beta sheets, which you don't see as well at low resolution, which is why they look more disordered. And for reference, this is the general architecture of the receptor we expect to see. Okay. All right, so let's come back a little bit to some classic pharmacology. We want to have our ducks in a row with function for these receptors, so we do some binding assays, an assay I learned in Palmer's lab, a simulation proximity assay for measuring affinity in solution. So we're looking at dissociation constants for ligands in detergent solubilized micelles. All right. So we have tritiated epibadidine. We can measure binding constant for it, and we see a KD of about high picomolar and no cooperativity in binding, consistent with what you see in rat brain receptors for the same type. The affinity is within or close to the published range, and to me it's surprising that it's even that close because our binding assays are done in detergent solubilized protein, and the assays in literature are all done from membranes. So I think that's close enough. We can compete off tritiated epibadidine with nicotine, which is what we've crystallized the receptor in complex with, and we can measure an affinity here, a Ki of 20 nanomolar, which again is very close to the published range for this receptor. Okay. This receptor is not just a binding protein, it's a ligated ion channel, so we need to show that binding of neurotransmitter actually promotes channel opening. So we transfect HEK cells with our subunits, and this is what we see with the wild-type receptor in whole cell configuration. We see fast activation, extensive desensitization, and then recovery when we wash off the agonist. And the crystallization construct is qualitatively indistinguishable in terms of function from the wild-type receptor. So that was a huge relief. Okay. All right, so wrapping this up, get to our break. I've shown you we've developed some new methods for virus titration and measurement of heteromer stoichiometry. Shown you some proof of principle on a nicotinic receptor. And now we have the raw material, we have a lot of protein, and hopefully soon we'll have structures to test mechanistic hypotheses related to the interplay between stoichiometry and structure and function. Okay, and then I will just say thank you to the people who actually did the work. This is my new lab at UT Southwestern. The people who did the work was Claudia Morales Perez, who was my first PhD student, and Colleen Noviello here in the audience. So thank you very much.
And again, we have time for a couple of questions. Steve?
Very nice. Right. It's a two-part question. What was your final stoichiometry?
2 alpha 3 beta.
2 alpha 3 beta. Second, with that stoichiometry, it should be distinguishable by attachment, electrophysiology, both in conductance and kinetics.
No, that's something we're missing, and it's a really we need to do that experiment either by getting EC50s for ligands to show that it is what it should be, and hopefully we can find a way to really robustly make that second stoichiometry and show that there's a difference between them, which would be a complementary validation of the method that I think is important to have. And binding assays, same deal. Yeah.
How do you think nicotine is increasing the production of the receptor?
Overall?
Yeah.
So I think it's just a general thermodynamic stabilization. So the receptor, the free energy of the receptor favors binding of ligand. The receptor is more stable when ligand is bound. And so when you have ligand around, the receptor is more stable, so it's easier to assemble as a pentamer. That's it. Yeah. And then the stoichiometric selectivity of stabilization comes from the same principle. The 3 beta 2 alpha form has a higher affinity than the other stoichiometry, and so that form is selectively upregulated because the binding energy is higher. Yeah.
And this would be selectively acting on a monomeric something on a subunit?
No, I think it helps to assemble specific interfaces. Yeah.
One last question from Jean-Pierre.
You showed 2 crystal forms, agonist and antagonist,
Yep.
which is of course extremely interesting. And you showed us one beginning of structure at phaton spread resolution. Now, have you the 2?
No. We see consistently better diffraction from nicotine bound, better than all other agonists and antagonists and combinations of agonists and modulators and channel blockers. And I don't understand that at all because there are much higher affinity ligands than nicotine. So we're pushing hard on nicotine right now. We don't have useful information for antagonists bound yet, but it crystallizes readily, so it should be doable.
In the case of the bacterial receptor, the agonist open state is highly resolved and has much less variability
That could be it, that could be it.
It's a sign of some kind of heterogeneity.
Yep, structural heterogeneity in the resting state. That makes sense.
Maybe.
Yeah.
So we have about 15 minutes for a break. Let's thank Ryan again.